Okay, so I started with the plasma membrane because that's the membrane that surrounds a living cell, so that's the basic structure of this lab. And then I went on to do vesicles off the plasma membrane, which with vesicles it'll be the fluid mosaic model because this has to do with fluid, like fluid molecules, and vesicles are fluid-filled like sacs. And then select permeability, where the membrane chooses what goes in and out of the membrane, like how it chooses the different like structures. And then active transport, which is going to be the definition of active transport is when the solution goes in through the membrane using ATP. And then off plasma membrane I use phospholipid bilayer, which is the two layers of the phospholipid, which you have a hydrophilic head of the phospholipid, which loves water, so it attracts the water, and then the hydrophobic tail of the phospholipid. And then amphiphalic molecule has to do with both of hydrophilic and hydrophobic, and then off the hydrophobic is the detergent molecule, which is attracted to water. And then the sandwich molecule, which has to do with the molecule with the sandwich molecule has to, or model, not molecule, sandwich model, LOL, describes the phospholipid bilayer. And then the passive transport is just freely transporting the solutions in between the membranes. And then concentration gradient is off of the passive transport because the passive transport has to do with a different concentration, and the concentration is where it's going to go from high to low in between the membrane. Osmosis is where it's like, for example, like fighting upstream. So like if you're trying to swim upstream, that's what osmosis does. It does from the lowest concentration to highest concentration. Isotonic is basically an equilibrium solution. And then hypotonic is going to be is a solution that has a lower osmotic pressure than another solution, and this one's just opposite of that. The gated ion channel is where it has it's like where it only allows certain ions in. And then electrochemical gradient is where it has to do with like the electrical charge of stuff. Membrane protein, we did the receptor membrane protein just because that's a type of membrane protein. Same thing with the peripheral membrane protein, transporter membrane protein, and the integral membrane protein, which off of that I have the ion channel because the ion channel allows for the integral membrane protein to accept ions, and the simple diffusion, which is going to be like I don't know. Simple diffusion is just going to be like, sorry, I get really sidetracked. Just going to be the diffusion like with no help that has to do with it. And then a protein attached membrane associated protein, then I put glycoprotein off the side of it. And then the lipid linked membrane associated protein, which then I put glycolipid connected to this. And then with the lipid, you'll have saturated fatty acids, and then the unsaturated fatty acids. And the unsaturated fatty acids creates the sterols and steroids. Here's my picture